Hi guys, I am Sajid Ali Shah. You are welcome to the Sun Biotechnology Association. Today we will discuss the dengue virus, genomic insights, its pathogenic mechanisms, and therapeutic approaches. So first we have to introduce the dengue virus. The dengue virus caused a most common disease, dengue fever, which is an arboviral disease. The dengue virus belongs to the family Flaviridae and the genus Flavivirus. It is mainly transmitted by the Aedes mosquitoes, by the Aedes aegypti, and up to some extent by the Aedes albicans. It is consists of five serotypes. The fourth are more common. For example, DNA 1, 2, 3, and 4 are most commonly found, and the DNA 5 was first reported during 2007 outbreak. The disease caused by one serotype doesn't produce immunity against the other serotypes. It means that during an outbreak, when you cure from the infection which is caused by DNA 1 or 2 at, at the same time, other type can also infect you and cause infection in you because the first one infection doesn't create immunity against the other serotypes of the DNA. Different terms are being used for the dengue fever and different ages. For example, a Somali term Chitinga eosphera derived from the word dengue, which means that fastidious or cheerful. Another term, breakburn fever, was used in an article during 1789 in the United States. Ballisamatic fever, dandy fever were also used by certain members around 1800, while the term dengue was used in English literature in 1828. So now we have to discuss types of dengue infections. How many types of dengue infections are there? So the dengue virus infection mainly divided into two categories: asymptomatics and symptomatic. The asymptomatics are those infections which mostly cure by itself and it doesn't produce any issues, any symptoms, etc. While the symptomatic infections are further divided into four categories: the undifferentiated fevers, dengue fever, dengue hemorrhagic fever, and dengue shock syndrome. Undifferentiated fevers are mostly non-specific and febrile illnesses are caused in it. While the dengue fever, it mostly affects the older children or adults. It causes high fever, retroorbital pain, myalgia, arthalgia, rashes, etc. While the dengue hemorrhagic fever that mainly consists of three phases: febrile, critical, and convalescent. While the dengue shock syndrome is the most severe form of dengue in which mostly cause death of the patients also. Now we will discuss the epidemiology. So from 2000 to 2019, the cases of dengue infections increased from 0.5 million to 4.2 million in 2019. It means that during the last 20 years, 8 times increased occur in the dengue infection. During the COVID-19 pandemic, an increase occurs in the incidence of dengue fever. In Pakistan, more than 22,000 dengue fever cases were reported in 2007, followed by 3,200 cases in 2018 and 24,547 cases in 2019, 3,442 cases during 2020, and 48,000 cases in 2021. While from January to 27 September to 2022, a total of 25,932 confirmed dengue cases and 62 deaths were reported in Pakistan. The first case of dengue was reported in Pakistan during 1882. After that, different pandemics occurs in different ages. For example, in 1994, 95, then 1998, and after that 2003, 2005, 2006, and then 2007, 8, 9, 11. Different pandemics occurs in Pakistan during the infection of dengue. Until now, about 1,57,485 cases were reported in Pakistan, and all and about 11 100 1153 patients have been died due to the dengue virus in Pakistan until now. Now we will discuss the structure of dengue virus. So the structure of the dengue virus is mostly spherical in shape. It consists of a single positive sense RNA and which is enclosed in capsid proteins, and the protein can also enclosed in a membrane. So the DNV is enclosed a tiny spherical virus which is positive single-stranded RNA genomes. The DNV serotypes in 1, 2, 3, and 4 shares almost 70 to 80% amino acid similarity. The dengue virus shares genomic and structural characteristics with other flaviviruses, which includes Japanese encephalitis virus, yellow fever virus, tick-borne virus, and West Nile virus. Now the genomic organizations of DNV. The genome of DNV is a single-stranded positive sense RNA, which is about 11 kb in size. It it's both in consists of the untranslated region 3' UTR and pipe UTR regions, and the other consists of structural genes and non-structural genes, which codes different proteins which perform different functions during the replications and attachments of the dengue virus. Genuine proteins and its functions. So the structural genes codes for structured protein, while the non-structural genes codes for the non-structural proteins. So the structural genes which were CPRM involve protein and membrane protein. The structural proteins are capsid proteins, which is about one 114 amino acids, while the involved protein is consist of 495 amino acids, and membrane protein is consist of 160 amino acids. While the non-structural regions of the genome codes non-structural proteins, which are NS1. NS1 consists of 352 amino acid, NS2A 218 amino acid, NS2B 130 amino acid, NS3 619 amino acid, NS4A 127 amino acids, NS4B 249 amino acids, and NS5 consists of 899 amino acids. These proteins perform different functions during the viral entry and its replication. For example, these are the proteins and these are their functions which they perform in the body. For example, the pre-membrane protein FRM perform it can target of antibody. It is the target of antibody response. It helps in infection enhancements, viral entry, viral assembly, and stabilizations of proper and the stabilization of proper folding of the E proteins. While the capsid proteins, it helps in RNA encapsulations in capsulations, viral packaging and release, viral growth and replications. The involved protein helps in cell attachments, fusion, antigenicity, immunological epitope, and infectivity. While NS1 helps in RNA replication, the NS2A it helps in antagonizing the host immune response, viral assembly, synthesis of viral RNA, inhibition of interferon signaling, formation of viral virions, and maturations. The NS2B helps in it performs the RNA triphosphatase, helicase, and protease activity, while the NS3 helps in polyprotein processing and unwinding and replication of RNA. The NS4A helps in interferon signaling inhibitions. The NS4B helps in interferon signaling inhibitions and viral replication, while the NS5 it helps in nuclear localizations, RNA polymerase, RNA capping, RNA polymerases, RNA capping, initiation of de novo RNA synthesis, and methyl transferase activity. It also perform a role in innate immunity and the infection and in the induction of interleukin 8, while the untranslated region, which is 3 UTR and 5 UTR. 3 UTR helps in RNA synthesis, replication, and host specific factors, while the 5 UTR helps in RNA synthesis and genome translation. Now we will discuss the life cycle of dengue virus. Now we will discuss the life cycle of dengue virus. So the life cycle mainly consists starts from the biting of Aedes aegypti or Aedes albicans, which contains the dengue virus. When the dengue virus enters into the body, it moves and attaches to the host cell. So there are certain receptors on the host cells for the attachments of these viruses. Some receptors are specific for the serotypes, while some are common to which all serotypes can bind. For example, Herefin sulfate, NLC4CR, DC, SIGNL, MNOS, etc. are the receptors to which all 1, 2, 3, and 4 serotypes can bind, while the receptors which are specific only for DNV2 are CRP78, CD protein associated proteins, unknown protein, trypsin resistant proteins. These are the proteins to which only DNV can bind, while there are certain proteins to which 1, 2, and 3 DNA can binds, which are the unknown protein, trypsin sensitive, and laminin proteins. While there are some certain receptors to which 2, 3, and 4 can bind, which is known as unknown proteins or serotype specific binding. So, firstly, mosquito comes and bite a human. Then the virus goes and attach into these receptors and then absorb into the cytoplasms. Then fusions of the virus occurs and it releases an uncoating of its take place. It releases its genomic RNA into the cytoplasm, to which the genomic RNA form other single-stranded RNAs, and some of the genomic, i.e. the RNA, moves into the endoplasmic reticulum, and here its translation takes place, and here it produce the structure and non-structure proteins. Same the process continues and a lot of proteins and single-stranded RNAs are formed. Then these combines together and assembled with each other. Polyprotein processing take place. The synthesized proteins have been processed and the assembly of the proteins and viral RNA take place and immature viruses are formed in the endoplasmic reticulum. These move to the Golgi complex where it matures and moves toward the cell membranes and released into the bloodstream where it moves and binds to other host cells and replicate inside them and infect these cells. In this way, life cycles of dengue virus completed within the host cell, and one virus come and it's performed a lot of viral copies in the host cell, and then these copies released from this cell and infect the other cells in the body. Now, pathogenesis of the dengue. So it is a complex interactions between the host gene, virus, and host immune responses. Host variables such as anti-DNV NS1 antibodies, memory cross-reactive T cells, autoimmunity, antibody-dependent enhancements, and genetic diversity are important contributors to disease susceptibility. These terms, these phenomena, these cells can affect the pathogenicity of the disease. There are also some genetic factors which also affect the pathogenicity. For example, the HLA-A32 and HLA-AA0202 alleles are when present in host in human. These are the protective phenotypes. Either the pathogenicity of the virus will be very low, while in case when HLA-A24 and HLA-A0207 phenotype when present are present, these are the harmful genetic variations. In those individuals, the pathogenicity of the disease will be very high. Mean it causes a very severe disease in those organisms. The pathogenicity mainly depends on the genetic makeup of the organisms, its anti-DNA NS1 antibodies, memory cross-reactive T cells, autoimmunity, and the antibody-dependent enhancements. Now the transmissions of dengue virus. The dengue is mostly transmitted by the mosquitoes known as Aedes albicans and Aedes aegypti. It can also transmitted from an infected person to the other ones through blood transfusion organ transplant, and maternal transmission. There are two cycles for transmissions of dengue virus. The one is jungle cycles in which the virus moves to the primates, for example, dogs, monkeys, etc., and then it can the mosquito bites these infected primates, i.e., dogs or monkeys, etc., and then bites the other uh, healthy ones and cause disease in those organisms. While the other cycle is urban cycles in which the virus is transmitted from one infected individual's person through Aedes mosquitoes into the other healthy ones, and this is known as the urban cycles of dengue virus transmission, and the other one is jungle cycles. While the virus can also be transmitted from an individual infected humans individual to the other one by direct blood transfusions, organ transplants, or maternal transmissions from mother to their offsprings. Now, what are the symptoms of dengue viruses? So, the symptoms are mainly consist of febrile phase, critical phase, and recovery phase. After the biting of the mosquitoes, after 7 days in the incubation periods, and in those days, no symptoms appears in the individuals or patients. But after that, viremia condition is created mean the virus amount increases in the body and febrile phase started from day 0 to day 7. In febrile phase, the symptoms will be headache, fever, pain behind the eyes, mouth and nose, bleeding, muscles and joint pain, vomiting, diarrhea, rash, etc. After the viremia stage, the non-viremic stage has been produced in which the viral loads decreases in the body, and this is mainly consist of critical phase and recovery phase. The critical phase lasted from 1 to 2 days, while the recovery phase lasted from 3 to 5 days, and after that the patients will recover. So the critical phase, in the critical phase the patients will cause hypotension, pleural effusions, gastrointestinal bleeding, and ascites. While in the recovery phase, there will be seizure, altered mind, and slow heartbeat. So these are the symptoms of dengue virus which will be felt during different stages of the disease. Now diagnosis or detections of dengue virus. So how we will detect or diagnose that it is a dengue infection or not? There are certain parameters which will be used for this. For example, we can detect it from blood parameters, blood tests. For example, we study or count the platelets in the body and HP. When the amount of platelets or HP decrease in the body, so we will say that the patient has dengue infections. After that, we will detect it through ELISA, IgG or IgM will be detected in the body. We can also use neutralization assays, nucleic acid amplification test. For example, we can use the polymerase chain reaction for detection of DNV. After that, we can also use biosensors and microarrays techniques. After, for example, when a mosquito bites from day -4 to day -1, we can't detect it. But from -1 up to day 5, it is a viremia stage, and in that stage, we can detect the virus by PCR. In PCR, we will detect the NS1 antigen of the dengue virus. While from day 0 to day 5, there will be fever, and we can also detect it through ELISA antibody detection. We can detect the IgG or IgM antibody in the body through ELISA techniques. But after day 4, risk of shock and hemorrhagic from day 4 to day 6, there will be the risk of shock and hemorrhage. So after the second day, we can detect the IgM in the body, but but after day 6, we will detect the IgG amount will be increased in the body, and after day 8, the amount of IgM will be decreased slowly in the body of the dengue infected patients. Now, prevention. How we will prevent ourselves from the dengue virus infection. So the first one, we should have to eliminate the standing water, either it is in our home or outside the home. We have to eliminate the standing water. Second one, we have to use the mosquito repellents. I have to use different types of repellents available in the market. We have to use this. We have to stay indoors. We have to stay indoors and don't come out from our homes to the jungles or grasses, etc. We have to wear protective clothing. We have to wear such type of clothes in which cover our whole body and the mosquito doesn't able to bite us. The other one is we have to use the mosquito nets, which will protect us from the mosquito biting during sleeping time. We can also use different types of mosquito control techniques. We have to use insecticide spray on homes. We have to use the laboratory engineer mosquitoes. We have to use such type of cloths which cover our whole body. Mosquito netting. We have to apply different types of insect repellents. Now, how we will treat the dengue infection? So, during the dengue infection, we have to do complete bed rest. After that, we have to maintain the fluid in the body. We have to use plenty of juice and we have to inject infusions into our bloods. In this way, we will maintain our body fluid because a lot of fluid lost during the dengue infections. Different plants have been used for the treatment of dengue virus in different ages. For example, Euphorbia herita and Quercia papaya, various Lucidinica, and other extracts of lemon leaf extracts and methonic extracts of plant Andrographis peninsula, Ocimum senatum, and these other plants have been used in different ages for the treatment of dengue virus infections, and they proved fruitful. For example, they help in the cure of dengue virus infections. Different articles have been available on the use of these plants for the treatment of dengue virus. We can also use different types of corticosteroids. We can also use antiviral drugs, i.e. protease inhibitors, ST148 and nucleoside inhibitors, vinazolic inhibitors, ST610, and immunosugars. We can also use the immunosugars. We can also use acetaminophen and for the treatment of dengue virus infections. After that, we can't use aspirin or ibuprofen during dengue infection because it has high side effects and dengue. Because the aspirin can decrease the thickness of blood and the loss of blood occur during the dengue infection. So you will lose mostly blood from the body when you use aspirin or ibuprofen. So the use of aspirin and ibuprofen were strictly prohibited during the dengue infections. We have to use calcium along vitamin D supplements during dengue infections. For the first, we have to do complete bed rest and but we have to maintain our body fluid during the dengue infections. Now, vaccines. So, vaccines are in different trials. Different vaccines have been passing from different trials for the treatment of dengue infections. There are certain vaccines which is used in different ages by the different people. For example, Dengue Vexa CYD-TDV, which is a live attenuated trivalent yellow fever vaccine used in Mexico, Thailand, Brazil, EI, Salvador, and Costa Rica. These vaccines have been used in these countries, while there are also currently some vaccines are in their phase 2 studies, which are the Tigdia tetravalent dengue vaccines. That is in the phase 2 trial of his productions. While two dengue vaccine TB003 and TB005 and DNVax are now being tested for effectiveness in Latin America and Asia. These are the different types of vaccines which is in different trials of their production. For example, live attenuated vaccine, chimeric live attenuated vaccine, inactivated vaccine, the subunit vaccines, and nucleic acid based vaccines. These are the different types of vaccines on which research is continuous, and inshallah they will be used in future for the preventions of dengue virus infection. Thank you. It's all from my side.